So it's my pleasure to introduce our second speaker today, who is Dr. Yun Gi, who is a postdoctoral researcher in the Wu Lab at Harvard. And Yun did her PhD in chemical biology at Peking University, where she was working on genetic code expansion and bioorthogonal chemistry. And in addition, at the same time, she was working on the evolution and engineering of enzymes and developing new intracellular proximity labeling approaches for detection of cell-cell interactions. Dr. Gi joined Christina in 2018 at the lab at Harvard, and since then she's been studying O-GlcNAcylation and devising new chemical biology tools to repurpose the targeting of the pair of enzymes OGT and OGA. So Yun, the floor is yours.
So thank you for having me today. It's my great pleasure, and I'm very excited to talk about my recent work on target protein deglycosylation in living cells by nanobody-fused split OGA. To start, I would like to briefly introduce O-GlcNAcylation. It's a monosaccharide post-translational modification on thousands of nuclear cytoplasmic proteins across species. As you can see here, nearly every major metabolic pathway is involved in the hexylamine biosynthetic pathway and regulates the end production of UDP-GlcNAc. As a high-end donor substrate, it can be transferred onto a serine threonine residue by a single enzyme OGT or removed by a hydrolase OGA, making O-GlcNAcylation reversible and highly dynamic. So O-GlcNAc modification tend to share the same functions among substrates and plays significant roles in a broad range of cellular processes, such as signal transduction, transcription, and protein stability. It also interplays with other PTMs like ubiquitination and phosphorylation. You may also observe aberrant O-GlcNAcylation in many diseases as well. Therefore, understanding where O-GlcNAc modification happens and what's a unique function of this PTM, especially in an individual protein, is what we are pursuing. So in order to address these questions, many methods for modulating O-GlcNAcylation have been reported, such as tuning the concentration of UDP-GlcNAc by interfering with nutrient metabolism. More commonly used approaches are targeting OGT or OGA by genetic modulation or inhibitors. However, these methods cause global perturbations on thousands of substrates and even irrelevant pathways. And treatments of inhibitors were reported to induce abnormal changes of endogenous OGT and OGA level. If we aim to study distinct O-GlcNAc functions on an individual protein, there are only a few methods we can apply, such as site-directed mutagenesis to achieve O-GlcNAc installation and removal permanently. But these all require identification of glycosides first, which remains challenging so far. In some cases, O-GlcNAcylation and phosphorylation may occur at the same or nearby site. So mutagenesis also brings a concern of disrupting potential crosstalk among PTMs. Therefore, we sought to devise a new approach for editing O-GlcNAcylation in a protein-specific way and facilitate the functional study. To achieve protein selectivity, the first tool that came to our mind is the antibody. However, canonical antibodies are too large to penetrate the cell membrane and unable to be expressed in living cells. Therefore, we turn to focus on another intriguing antibodies generated from camelids. Unlike canonical antibodies, the candidate antibody only contains a heavy chain and is able to bind the antigen as a single domain, which is called a VHH or nanobody. So given its small size, high affinity, and good biocompatibility, nanobodies have been applied in many fields. So we envision that nanobodies are suitable to target O-GlcNAc modified proteins inside living cells. So we first choose GFP and the well-characterized nanobody against the GFP at the recognition pair. GFP nanobody will selectively bind GFP, which is fused with the target protein like NUP62 here, and thereby recruit the tethered OGA around to remove O-GlcNAc from NUP62. The crystal structure of OGA shows that it forms a unique arm-in-arm homodimer by swapping the stop domain for maintaining activity. More intriguingly, OGA was reported to be cleaved by caspase-3 during apoptosis, and the two fragments can still bind to each other and reconstitute enzymatic activity after cleavage. We verify this feature on NUP62 by expressing two fragments simultaneously in living cells. According to the O-GlcNAc plot, we observed sugar removal on NUP62 by the two fragments as expected. So we took the motivation from this feature and decided to split OGA at the cleavage site and screened different truncations of N fragments and C fragments for a lower association pair, so that lower inherent activity. By testing activities of different combinations on GFP NUP62, we found that a shorter N2 fragment can still help regain the activity and deglycosylate GFP NUP62 when paired with the original C1. We next screened C fragments in combination with N2 and found that the shorter C fragment is the less active the split OGA is. We then wondered upon installation of the nanobody, whether the sugar removal activity of the last two less active split OGA combinations could be restored towards the target protein. From the O-GlcNAc plot, we're excited to see that the split OGA shows little activity on GFP NUP62, but upon tethering nanobody on either N2 or C3 fragment, it regains the deglycosylation ability on NUP62. However, no activities was observed with the N2 and C4 fragment, C4 pair after fusion of nanobody. Given the results of further comparisons, we decided to use N2 and the nanobody fused C3 as our protein selective O-GlcNAc eraser. Then we started to comprehensively characterize this tool, especially in terms of its generality and selectivity. So in addition to GFP NUP62, we tested the nanobody-splitOGA on another 4 glycoproteins spanning highly and mildly modified substrates, and confirmed that splitOGA alone is not active enough to remove O-GlcNAc from, for example, SP1 here. But fusion to nanobody will regain its activity on the target protein and achieve selective sugar removal. Besides, unlike split OGA, nanobody fusion co-localize well with the target protein SP1 without changing its nuclear localization, indicating the great targeting ability and the minimal perturbations of nanobody split OGA. We also applied a mass shift assay to quantify O-GlcNAc modification level on another target protein, GLP-1B, and a non-targeted endogenous glycoprotein, CRAB, simultaneously. As we expected, both CRAB and GLP-1B can be equally deglycosylated by Fordham's OGA. However, nanobody-splitOGA can only selectively remove O-GlcNAc from targeted GLP-1B instead of the non-targeted CRAB, implying the good selectivity of this eraser. Furthermore, we evaluated the selectivity of this O-GlcNAc eraser from a global proteome perspective. Using chemoenzymatic labeling and quantitative proteomics, we enriched and quantified the O-GlcNAc modified proteins under the treatment of different OGA constructs. From the volcano plot, we can see that the global O-GlcNAc proteome in the presence of split OGA, including our target protein GLPSP1, showed no significant difference from that seen with the inactive form, indicating that split OGA alone has little global perturbation. However, for comparison of cells co-expressing nanobody split OGA to cells co-expressing split OGA, the target protein GFP-SP1 displayed the largest O-GlcNAc reduction with the lowest p-value, indicating the great activity and selectivity of our nanobody split OGA. As treatments of OGT or OGA inhibitors were reported to induce upregulation of endogenous OGT and OGA level respectively, we wondered if overexpression of nanobody split OGA would break the homeostasis of endogenous OGT and OGA level as well. So in the presence of different OGA constructs, we didn't observe significant changes on endogenous OGT and OGA levels, suggesting that expression of a nanobody split OGA didn't break the homeostasis balance, which is what we expected. As GFP tagging may not be ideal under all circumstances for all substrates, we next evaluated other nanobody tag pairs, in particular those small peptide tags such as a C-terminal EPEA tag, a 14 residue UBC tag, and a BC2 tag. All these nanobody tag pairs are compatible with split OGA system and successfully remove O-GlcNAc from target protein NUP62 or another glycoprotein c-Fos. The deglycosylation ability may vary a little bit due to different binding affinities of this nanobody against corresponding tags. At last, we want to apply this tool on a target protein to decipher the unique functions of O-GlcNAc. Through a mass shift assay, we first demonstrated that nanobody split OGA is able to selectively erase O-GlcNAc modification from a transcription factor c-Jun. Then we monitored the turnover of GLP c-Jun by adding CHX to block protein synthesis and found that it degrades faster with the treatment of nanobody split OGA rather than the inactive form, indicating that the loss of O-GlcNAc on GLP c-Jun impairs protein stability directly. c-Jun also belongs to AP1 transcription factor complex by dimerizing with c-Fos for DNA binding. And I just showed that O-GlcNAc modified c-Fos can be selectively deglycosylated by nanobody split OGA in previous slide. Therefore, we sought to explore a possible connection between O-GlcNAcylation and AP1 transcription activity. As we can see here, upon the treatment of OGT inhibitor osmi-4B, we observed a dramatic reduction of global O-GlcNAcylation and an enhanced AP1 transcription activity. However, the transcriptional activity remained unperturbed upon selective c-Fos deglycosylation using nanobody split OGA in comparison to the inactive mutant, indicating that direct deglycosylation on c-Fos does not promote AP1 activity. We postulate that the enhanced transcriptional activity induced by OGT inhibitor may be promoted by removal of O-GlcNAc from many other proteins and is not directly correlated with O-GlcNAc modification on c-Fos. Given these results, we believe that nanobody-splitOGA would help build the direct connection between downstream phenotypes and O-GlcNAc modification on a target protein, so that complement the use of OGT inhibitor for O-GlcNAc study. To summarize this work, I have designed the first protein selective O-GlcNAc eraser by leveraging the selectivity from nanobody and the enzymatic activity from OGA. The nanobody-splitOGA we design is modular and generalizable, enabling the dissection of O-GlcNAc functions on a protein of interest. And this approach doesn't require glycosides information or cause large global perturbations as well. And we have already initiated some collaboration, but welcome more to apply this eraser on more or many other complicated biological systems. In the end, I would like to thank Christina and people who have contributed to this work, and also lab members in O-Lab for being a very supportive team. And thanks for all these fundings and organizers for having me today. And I'm happy to take any questions.